The urinary system is responsible for filtering blood, processing that filtrate, and eliminating the waste. It makes sense then that the urinary system would also be involved in regulating the content of our blood. We will be looking at these processes, how they are controlled, and how we use the urinary system to regulate blood osmolarity. Some physical characteristics of urine. Color. It should be clear to deep yellow depending on water content and diet. For example, first morning urine is concentrated and darker in color. Later in the day, once you've hydrated, urine is much lighter in color. Certain vitamins and herbs can affect color, such as the case with turmeric found in curries. Whatever the color, urine should never be cloudy. That's an indication of infection, and it shouldn't be red or dark brown. Odor. Fresh urine has little odor, but if allowed to sit for a while, it will begin to smell of ammonia. Again, diet can affect odor. Some people metabolize an amino acid found in asparagus into a volatile, smelly compound that comes out in the urine. Vitamins can have a similar effect. pH. Urine is one way we excrete acid or base from the body. Because of that, the pH of urine can vary widely. And again, diet can have an effect on this. Specific gravity. Note the small range. This is a measure of solute concentration in urine. It is tightly controlled as it does affect blood osmolarity. And miscellaneous. Again, we see different chemicals getting removed from the body through the urine. Urea, uric acid, and creatinine are waste products found in predictable amounts. We also see various amounts of ions being excreted. A list of renal functions, some of which we will cover in detail. Filtration. That's allowing the plasma and small solutes to leave the blood. This is pressure gradient controlled. Regulation, obvious, using renal functions to stay in homeostasis. Osmolarity, hopefully obvious, solute concentration regulation. Vitamin D activation, not so obvious. Vitamin D is made by our skin, but activated in the kidneys. That's all you need to know about this one. EPO. Remember erythropoiesis? Review that content. Blood pressure. Remember the renin-angiotensin pathway and short and long-term controls over blood volume? Review that content. Gluconeogenesis. Something new. The ability to cause glucose to be made by the liver from molecules that were not carbohydrates. That's all you need to know about this one. The functional and structural unit of the kidney is called the nephron. It consists of several parts. 1. The renal corpuscle. It contains a capillary called the glomerulus, as well as the first nephron structure that's wrapped around it. That is called the glomerular capsule. Renal tubule. Attached to the glomerular capsule, it is a long tube that has specialized structure and function along the way. In order for the glomerular capsule, the renal tubule consists of, in order from the glomerular capsule, the renal tubule consists of the proximal convoluted tubule, loop of Henle, distal convoluted tubule. The renal tubule then empties into a larger collecting duct, which flows the waste liquid through a papillary duct into the cavities of the kidney. A diagram showing the specific structures of both the renal corpuscle and renal tubule. Why names for these two things? Because the structures of the renal corpuscle perform filtration. However, the structures of the renal tubule perform reabsorption and secretion. In other words, the renal corpuscle filters our blood, but the renal tubule processes that filtrate. You will still need to know the individual structures and their specific functions. Another diagram of the nephron structures. Here we see the glomerulus as a capillary coming off the red blood vessels. We can also see that the tissue type of the renal tubule structure changes. Spend some time looking at this particular diagram. In fact, diagram it out in your notes. Let's zero in on the renal corpuscle structures: glomerulus, glomerular capsule. Remember, those structures perform filtration of the blood. The glomerulus is a specialized capillary bed that is fenestrated. That makes it very permeable to liquid and small molecules. It is also under high pressure, so there's always a force that is pushing the fluid of blood out of the capillary and through the fenestrations. This fluid, or filtrate, will then collect in the space between the glomerulus and the glomerular capsule. Now look at the glomerulus again. Note how there are yellow cells wrapped around the capillary. These cells are called podocytes, and they are part of the glomerular capsule. In fact, the glomerular capsule has anatomy similar to a serous membrane. A visceral layer of podocytes that is connected to the glomerulus, and an outer parietal layer forming a cavity. The podocytes don't wrap all the way around the capillaries, but have a space where they don't overlap. These spaces are called filtration slits. Filtration slits help us control the molecule size of what leaves the blood. We want water and small solutes to be removed from the blood. We do not want proteins or cells leaving blood. Filtration slits prevent the larger proteins and cells from leaving the blood. What would happen if the filtration slits were too big and proteins leave the blood and escape the body? In your response, you should include the following terms: plasma proteins, osmolarity, capillary osmotic pressure, and edema. Post your response to the question in the slide in the A&P Cafe. A diagram showing the filtration membrane. When fluid and small solutes leave the glomerulus and enter the glomerular capsule, they're traveling across this membrane. It is extremely thin, like all exchange membranes, and consists of the simple squamous endothelium of the glomerulus with its fenestrations, fused basement membranes, and the podocytes with their filtration slits. You can see here how the filtration slits are a little smaller than the fenestrations. Again, they help control the size of molecules that leave the blood during filtration. Imagine fluid in the plasma getting pushed out the capillary through the fenestrations, across the fused basement membrane, moving through the filtration slits, and entering into the capsular space. Let's now focus on tissue types of these structures. The glomerular capsule structures are simple squamous epithelium and perform filtration. The proximal convoluted tubule is composed of simple cuboidal epithelia with microvilli. They perform reabsorption of useful molecules, which pulls them out of the filtrate and returns them to the blood. They can also remove waste we did not catch in filtration and push these into the filtrate for removal. The loop of Henle has two limbs. The thin segment is simple squamous and performs osmosis, but the thick segment is simple cuboidal. That portion reabsorbs solutes to create an osmotic gradient. The distal convoluted tubule is simple cuboidal cells with no microvilli. Like the proximal convoluted tubule, it's reabsorption or secretion of different molecules. Take a few minutes to review the structures of the renal corpuscle and renal tubule, along with their tissue types and general functions. Recall that the nephron ends at the distal convoluted tubule. Whatever fluid is left in the lumen at this point is now called urine. Urine from multiple nephrons will now enter into the collecting tubule or duct, where there is one last chance to process it before it leaves the kidneys. Collecting tubules have two cell types: intercalated cells for acid and base balance, see how they pump bicarbonate and hydrogen ions, and principal cells for water and salt balance, see how they pump sodium, potassium ions under aldosterone stimulation. Regarding principal cells, where sodium goes, water follows. So it turns out that there are two types of nephrons. Most nephrons in the kidney are small ones, and most of their structures are in the outer cortex region. Because of this, they are called cortical nephrons. However, some nephrons have their corpuscle and convoluted tubules in the deeper part of the cortex region, but they have super long loops of Henle that are in the medullary region. These are called juxtamedullary nephrons. That word basically means next to the medulla. Since they have long, specialized loops of Henle, they are super good at osmosis and are responsible in part for pulling lots of water out of the filtrate to concentrate the final urine. In fact, we filter out over 150 liters of filtrate every day, but you'll only make about 6 to 8 liters of urine. That means about 150-ish liters of water are reabsorbed every day. Juxtamedullary loops of Henle help us do this. With all this filtering, reabsorbing, and secreting, where does all that stuff go? Well, kidneys have 3 different types of capillary beds for all this. The glomerulus in the renal corpuscle is for filtration and is fenestrated. It is specialized and is a higher pressure capillary bed because the afferent arteriole has a larger diameter than the efferent arteriole. At any moment in time, more blood enters the capillary than leaves. So blood backs up, and the pressure of this capillary bed stays high, unlike regular capillaries. 2. Peritubular capillaries surround the renal tubule. When the cells of the different structures of the renal tubule absorb useful molecules from the filtrate, they'll put those molecules into the blood using the peritubular capillary. If they pull a waste molecule out of the blood to dump it into the filtrate for removal, they will use the same peritubular capillary beds. 3. Vasa recta are specialized capillary beds of the loops of Henle of juxtamedullary nephrons only. They help maintain osmotic gradients with a specialized flow. More about that later. This diagram shows us the different capillaries. The red glomerulus for filtration, the purple peritubular around the convoluted tubules, and lastly, the purple vasa recta of the juxtamedullary nephrons loop of Henle. Last structure for now, the juxtaglomerular apparatus. You can see that it is associated with the renal corpuscle here. There are two components. Macula densa cells help control osmolarity. They're found inside the distal convoluted tubule, right where that passes between the two arterioles of the glomerulus. Those cells are monitoring the osmolarity of the filtrate that is flowing through the lumen at this spot. The second part of the JGA are granular juxtaglomerular cells. These surround the afferent arteriole, green on the diagram. The JG cells monitor how much blood is in the arterioles. They'll use this information to control how fast blood gets filtered, a filtration rate, and blood pressure. Finally, we are now ready to look at these processes. Don't get caught up in the details here, but we filter our blood constantly and produce a lot of filtrate throughout the day. That filtrate is processed so that useful molecules are recycled, but waste molecules are eliminated. Along the way, we will recycle a lot of water. The three processes of urine formation: glomerular filtration at the corpuscle, and tubular reabsorption and secretion at the tubule. Glomerular filtration occurs because of the pressure gradient between the glomerulus and the glomerular capsule. It is not specific, but sorts by size only. Whatever is small enough to fit through the filtration slits is pushed out of the glomerulus. Tubular reabsorption occurs in the tubules. This is very specific. The simple cuboidal cells in different parts of the tubule will move different useful molecules out of the filtrate, returning them to the peritubular capillary. Again, this process is recycling and is chemically specific. Tubular secretion. Filtration is not substrate specific, and waste molecules sometimes get missed. Secretion is our last chance to get rid of them. The simple cuboidal cells of the tubule structures can move waste molecules from the peritubular capillaries to the lumen of the tubule. This is also chemically specific. A diagram of the corpuscle. Remember, this is where filtration happens. Filtration occurs whenever we have a pressure gradient and a leaky capillary, which is exactly what we see here. There are three pressures involved: glomerular hydrostatic pressure, blood colloid osmotic pressure, and capsular hydrostatic pressure. Glomerular blood pressure. This is a function of blood pressure and the volume of blood inside the glomerulus. It is a force pushing against the walls of the capillary. So, in other words, this is a pressure pushing out of the capillary. It can be changed moment to moment simply by changing how much blood is in that capillary. Allow more blood in, the pressure increases. Allow less blood in, the pressure decreases. Blood colloid osmotic pressure. This is a function of plasma proteins in the blood. Water in the capsular space is attracted to these plasma proteins just like regular capillaries, and is trying to get back into the capillary toward those plasma proteins. So in other words, this is a pressure pushing into the capillary. It cannot be changed. Remember, it is a function of plasma proteins. The only way this changes is if we're in liver failure and can't make the plasma proteins, or in kidney failure and we're peeing out the proteins. Capsular hydrostatic pressure. This is a function of water in the capsular space as well. The water here is pushing against the capillary wall, creating its own force. But again, this is a pressure pushing into the capillary, and also it cannot be easily changed. When we look at these three pressures, we see one pressure pushing out versus two pushing in. As long as the pressure pushing out is greater than the pressures pushing in, some fluid will leave the glomerulus and collect in the capsular space. Remember, only the pressure pushing out can be changed moment to moment, while the pressures pushing in are constant. Well, how does filtration work? Well, blood enters the glomerulus, creating a pressure pushing out that is larger than the pressures pushing in. There will be a net movement of fluid with this gradient, or a net fluid of movement, or fluid will move out of the capillary into the space. One more time. There will be a net movement of fluid with this gradient. Fluid will move out of the capillary into the space. If we allow more blood into the capillary, we will increase the gradient, and even more fluid will get pushed out of the capillary. If we allow less blood into the capillary, we will decrease the gradient. Less fluid will get pushed out of the capillary. So really, filtration is a battle between pressures. Filtration occurs because of pressure gradients across a highly permeable membrane. Let's say this in regular words first. The pressure pushing out controls the gradient, and the gradient controls the speed at which we filter. If you change the pressure pushing out, you will change the gradient. And if you change the gradient, you will change the speed in which we filter. Repeat this about 10 times until you can do it by yourself. The pressure pushing out is the glomerular blood pressure. The pressure gradient is net filtration pressure. The speed in which we filter is glomerular filtration rate. Now let's translate those regular words into our anatomy words. The glomerular blood pressure affects the net filtration pressure, and the net filtration pressure affects the glomerular filtration rate. Remember, large molecules are never filtered out. The formula for this concept: the net filtration pressure is a function of the pressure pushing out minus the two pressures pushing in. The speed in which we filter is the glomerular filtration rate. It is controlled by the net filtration pressure. The change is proportional. Increase the net filtration pressure, and we increase the glomerular filtration rate. Lower the net filtration pressure, and we lower the glomerular filtration rate. Filtration can be changed by modifying the volume of blood in the capillary. But it's also affected by total surface area for exchange and permeability of the membrane. Glomerular filtration rate is tightly controlled. If it is too fast, way too much filtrate will move through the tubule, and the simple cuboidal cells of the tubule structures won't be able to capture all the useful molecules. We will end up losing nutrients in our urine. If it's too slow, the simple cuboidal cells of the tubule structures will reabsorb everything, including waste. We'll end up having increased waste in our blood. We have to maintain a balance between too fast and too slow for moment-to-moment homeostasis. Since controlling glomerular filtration rate is important, we have lots of options. Autoregulation. This is intrinsic control at the renal corpuscle structures. This includes myogenic and flow-dependent mechanisms. The nervous system. Extrinsic control using the sympathetic nervous system. In hormones. Extrinsic hormonal mechanisms such as norepinephrine, epinephrine, and the renin-angiotensin pathway. Here's the deal. Regardless of how we control glomerular filtration rate, we have two basic options. Control how much blood is inside the glomerulus, or control how much blood is in the body. Myogenic, a property of the afferent arteriole. This arteriole controls how much blood enters the glomerulus. If too much blood is in the glomerulus, such that the glomerular filtration rate is too high, well, what makes sense? Leave it high or get it back down again. How do we get the GFR back down? Well, we have to lower the net filtration pressure. And how do we lower the net filtration pressure? Well, we have to lower the glomerular blood pressure. And how do we lower the glomerular blood pressure? By decreasing the amount of blood that enters the glomerulus. Well, and how do we lower the amount of blood entering the glomerulus? By constricting the afferent arteriole. The afferent arteriole will do this on its own. If extra blood stretches its wall, it responds by squeezing back to prevent all that extra blood from getting into the glomerulus. Flow-dependent tubuloglomerular feedback. Another way to correct a glomerular filtration rate that's too high. When the GFR is too high, too much fluid is moving through the tubule. Since it's moving so fast, there's not enough opportunity to reabsorb useful things like sodium. This means that there will be too much sodium in the filtrate. The macula densa cells of the distal convoluted tubule will detect the elevated sodium. In response, they'll release a paracrine, a local hormone, that travels to the afferent arteriole. And in response, the afferent arteriole will vasoconstrict. An overview. There's too much filtrate in the tubule, so you need to make less to get back to homeostasis. We make less filtrate by decreasing the amount of blood that enters the glomerulus. And we do that by constricting the afferent arteriole. A diagram of myogenic autoregulation and its sequence of events. A diagram of tubuloglomerular feedback and its sequence of events. the same time of all that intrinsic excitement, we will have extrinsic excitement. Thankfully, you know about these already. The sympathetic nervous system will vasoconstrict the afferent arteriole to decrease the amount of blood in the glomerulus, which decreases its glomerular blood pressure, which decreases the net filtration pressure, which decreases that glomerular filtration rate. Epinephrine and norepinephrine also constrict, and the renin-angiotensin pathway also constricts. A diagram of the renin-angiotensin pathway. When blood pressure is low, JG cells detect it and secrete renin. Renin converts angiotensinogen into angiotensin II. Angiotensin II does two things. One, it's a potent vasoconstrictor body-wide, so it directly increases blood pressure. And two, causes production of the hormone aldosterone from the adrenal cortex. Aldosterone travels to the collecting tubule and stimulates sodium reabsorption. Where sodium goes, water follows. So this is a sneaky way of stimulating water reabsorption. An overview diagram. Pause. Spend some time with just this diagram. It covers everything we talked about: myogenic autoregulation, tubuloglomerular feedback, renin-angiotensin pathway, and sympathetic nervous system. I recommend you print it out and study from it. So that's more than you ever wanted to know about filtration. Now, that filtrate is going to be processed by the structures of the renal tubule. Reabsorption happens when the simple columnar cells pull something useful out of the filtrate and dump it into the blood inside the peritubular capillaries. That means the cell is moving the molecule past its apical and basal surfaces, across the basement membrane, and then into the capillary. What moves? Organic nutrients, water ions. How are they moved? Active and passive transport. Again, another overview diagram. Don't get caught up in details here, but focus more on general processes. For example, passive transport of useful materials include osmosis of water and water-soluble solutes such as ions. Those same ions can also pass across the cell membrane, transcellular, in active or passive transport. Passive transport is facilitated diffusion, whereas active transport would be protein pumps. It turns out that sodium ions are useful to fuel transport mechanisms. We can pump sodium with the ATP-driven sodium-potassium pump. We can use sodium to help a carrier protein in the cell membrane move a molecule. For example, we move sodium with a glucose on the same protein across a membrane. We can even use it to transport chloride across a cell. Basically, where sodium goes, water and ions follow. Here we see the sodium being used to move other items across the simple cuboidal cells. You can see sodium move glucose in the same direction at the same time with the red dashed lines. That direction is called symport. We can also see sodium and potassium moving in opposite directions with the pump. That direction is called antiport. This picture also shows us osmosis of water, ions traveling with water between cells, and fat-soluble molecules moving across cells. However, polar molecules like amino acids, other ions, and vitamins have to travel on protein carriers with sodium. Once fluid moves through the tubule, whatever molecules are not reabsorbed will be left in the fluid. That fluid is then called urine and will be excreted. Turns out, all molecules that are reabsorbed and kept as useful molecules to be recycled have predictable reabsorption rates and values. The transport maximum is basically the maximal amount of a molecule that can be reabsorbed at any moment in time. Why do we care? Because at this point, any additional solute molecules cannot be reabsorbed and will end up in the urine. For example, a diabetic does not control their blood glucose well. They'll have excess glucose in their blood, so excess glucose in their filtrate. The proximal convoluted tubule will reabsorb glucose up to the TM, or transport maximum. That extra glucose will stay in the urine. This is why diabetics get glucose in their urine. And again, why do we care? Because where glucose goes, water will follow via osmosis. Extra glucose in urine means extra water in urine. That's exactly why diabetics urinate frequently. There are several factors that affect the transport maximum, or TM, of a substance. Carriers. Have the simple cuboidal cells made enough carrier proteins to move the substance? Solubility. Polar molecules have to move across carriers, but lipid-soluble nonpolar molecules are soluble directly across a cell membrane. And size. If the substance is larger, it has to be transported using vesicles. Lastly, the transport maximum of critical substances is clinically important because there are no normal values. Here we see urea, creatinine, and uric acid, to name a few. Again, don't get caught up in the details. We do need you to know that substances are reabsorbed in each part of the renal tubule, but there's a big picture here. Proximal convoluted tubule reabsorbs organic nutrient molecules. Loop of Henle reabsorbs water by osmosis. Distal convoluted tubule reabsorbs electrolytes, bicarbonate, and hydrogen ions. And collecting duct reabsorbs water under hormonal control. Remember, sodium's an important ion, right? We can use sodium to control substance movement. We especially use sodium to control water movement. So what makes sense to you if blood pressure and blood volume are too high? Should we leave it high or get it back down again? Hopefully you said get blood pressure back down again. We have lots of blood vessel ways to solve that problem, but we can also use the urinary system to help solve the problem. When blood pressure is high, we end up stretching the walls of the right atrium. In response, the atrium secretes a hormone called Atrial Natriuretic Peptide. ANP travels through the blood to our kidneys. Here, it acts on the collecting duct and stimulates sodium secretion. In other words, the collecting duct will move sodium out of the surrounding blood in its peritubular capillary and push it into the urine. Where the sodium goes, water follows. So really, ANP, or atrial natriuretic peptide, helps excess water be pushed into the urine. Over time, you'd end up peeing out the extra sodium in water, which will eventually lower your blood volume and pressure. Atrial natriuretic peptide also inhibits any sodium reabsorption during this time. It blocks angiotensin II, and it increases glomerular filtration rate. Answer these questions in the A&P Cafe. Answer these questions in the A&P Cafe. Tubular secretion. This looks a lot like reabsorption. The simple cuboidal cells are moving a substance across them, using either active or passive transport and carrier proteins. Only secretion is in the reverse direction. Waste molecules still in the blood within the peritubular capillaries are grabbed by the tubular cells and dumped into the filtrate so that you remove them from the body. Secretion, you can think of secretion as taking out the trash, whereas reabsorption is recycling. Substances that are secreted include urea, uric acid, excess potassium, and hydrogen ions for blood pH control. A basic diagram showing reabsorption, secretion, and filtration throughout the nephron. The yellowish structure is the nephron, but the pinkish structure are the blood vessels around them. And now it's time to focus in on the loop of Henle. We've talked about filtration at the renal corpuscle structures, reabsorption and secretion of convoluted tubules and the collecting duct, but we've yet to examine the loops of Henle. In fact, we're going to focus on juxtaglomerular nephron loops of Henle. These structures are extremely good at controlling water reabsorption. Again, why is that a big deal? Because the amount of water and solutes in our blood determines our osmolality, which in turn determines how fluid moves through our body's different compartments. Think capillary dynamics and edema. So we need a system to really do a good job at reabsorbing water and electrolytes. Now, milliosmoles is a measurement of solutes, and I'm about to show you a countercurrent mechanism. The loop of Henle of juxtamedullary nephrons is really good at removing water from the filtrate by osmosis. What happens is the loop will actively move salt from the filtrate into the interstitial fluid to create an osmotic gradient. If you look at the numbers and the kidney diagram, you can see the gradient. Near the cortex, we end up with a little salt in the interstitial fluid, and so the number is small, around 300 milliosmoles. As we move down into the medulla toward the sinus, we see the interstitial fluid salt concentration rising. It is the loop of Henle pumping salt into the interstitial fluid to create this gradient. Now contemplate this. One side of the loop of Henle is going to make the gradient by pumping those salts. The other side of the loop of Henle is going to use the gradient by allowing water to move by osmosis. Note, the loop and tissues in the medulla are alive. So do they need a blood supply? Absolutely. We need blood vessels to bring nutrients to and waste from the living cells and tissues here. That's one job of the capillary bed called the vasa recta. But unfortunately, this poses a problem. If we allow blood to flow into the area, do exchange, then flow out of the area in the old-fashioned way, that salt gradient the loop worked so hard to get would disappear. In other words, the salts in the interstitial fluid that we put there on purpose will diffuse into the capillary and be taken away by the blood. We would end up losing our gradient, and we couldn't then move water by osmosis, so we need to solve this problem. How do we do it? With a specialized capillary bed. The vasa recta you're looking at is not normal. Normal capillaries would move blood in the same direction as the filtrate is moving in the tubule. But this is not the case here. The vasa recta's blood flow is opposite to the direction of the filtrate. This opposite direction of flow is called a countercurrent mechanism. So instead of salts diffusing into the moving blood and being taken away, the countercurrent mechanism actually maintains the salt gradient. Here we take another look. The ascending loop of Henle is permeable to salts but impermeable to water. That means that the ascending limb cells actively transport salts from the filtrate to the interstitial fluid. This limb makes the gradient. The descending limb has nothing to do with transporting salts. Instead, the descending limb is permeable to water. Water will diffuse out of the descending limb according to the osmotic gradient. The top of the loop is near the cortex, and there's only about 300 milliosmoles, so a little water leaves the filtrate to the interstitial space, where it gets picked up by the vasa recta. As the filtrate moves down deeper into the medulla, it encounters a larger gradient of salts. At the end or bottom of the loop, there's about 1,200 milliosmoles of salt in the interstitial fluid. So even more fluid will diffuse out of the filtrate. By the time the filtrate reaches the little hairpin turn of the loop, tons and tons of water has diffused out to the gradient and been picked up by the vasa recta. So the descending limb uses the gradient. Lastly, remember that it's the job of the vasa recta to not only keep the tissue alive, but also remove the water. And because it is flowing in an direction opposite the filtrate, all concentration gradients are maintained. The vasa recta maintains the gradient. Again, ascending loop of Henle makes the gradient. Descending loop of Henle uses the gradient. Vasa recta maintains the gradient. Another view of the countercurrent exchange mechanism. In yellow is the loop of Henle. The ascending limb is pumping salt to make the gradient. The descending limb is allowing water to diffuse out according to the gradient. And the vasa recta is picking up the water flowing in an opposite direction so as to remove the water from the area. As a recap, we have learned the mechanisms and control of filtration, the general processes of reabsorption and secretion, and more than you ever wanted to know about the juxtamedullary loops of Henle and a countercurrent exchange system. Now we'll touch base on the last structures, collecting ducts. Collecting ducts either reabsorb sodium and therefore water, or secrete sodium. This is under hormonal control. The posterior pituitary can secrete a hormone called anti-diuretic hormone. When there's no ADH, the cells of the collecting duct leave sodium in the urine. This will leave water in the filtrate, and you'll have a dilute urine. This happens during the daytime, if you're fully hydrated, and even if you consume alcohol or caffeine, as they block ADH action. Now imagine what happens when you're dehydrated or during the night if you're sleeping. Under those conditions, ADH is released. It acts on the collecting duct cells and triggers sodium reabsorption. Water travels with it, and you'll end up producing a concentrated urine. This is why your urine is darker in color in the morning and why so many clinical tests request first morning urine. Any molecule you're testing for will be most concentrated at that time because you've been reabsorbing the water all night long. Once humans learned how urine is produced and how ions and hormones affect how much water is excreted or reabsorbed, they manipulated it with medication. A diuretic is anything that results in a dilute urine. In other words, a diuretic is anything that makes you pee a lot. Some diuretics will inhibit those sodium transports. Lasix and diurea work this way. And since you leave sodium in the urine, that means you'll leave the water in the urine. There are other diuretics like glucose that just mess with the osmotic gradients. Remember, where glucose goes, water follows. Anything that dilates the afferent arteriole of the glomerulus will increase glomerular blood pressure, which increases net filtration pressure, which increases glomerular filtration rate. That's why alcohol and caffeine both make you pee in addition to blocking ADH, and why they are dehydrating. Last new term. The time it takes to filter blood, process the filtrate, and excrete the waste for any given substance is called renal clearance. You don't need to memorize this formula. We do want you to know what it means. The renal clearance rate, or the speed in which you do everything we've learned about for any given molecule, is dependent on several things. 1, the concentration of that substance in your urine. 2, the speed in which you're making urine. And 3, the concentration of that same substance in your plasma. Here's an example. Asparagine is the amino acid in asparagus. Asparagine contains sulfur, and some people's livers will metabolize that amino acid into a sulfur-based waste compound. That sulfur compound ends up in your blood, gets filtered at the renal corpuscle, and is not reabsorbed. As a result, the sulfur compounds come out in your urine. The renal clearance is the amount of time it takes for asparagine's metabolite to be filtered and excreted. The more asparagine you eat, the faster this happens. Also, the more dehydrated you are, the more concentrated the compound will be and the faster it'll be cleared. So eat some asparagus for dinner tonight and experience your renal clearance rate. It's amazingly fast. Urine is constantly being made because you are constantly filtering. That process never stops. So urine is constantly dripping out of the sinus, into the ureters, and then into your bladder. But obviously, you don't constantly pee. That is because our bladders accept that constant drip of urine, but hold on to it until the bladder's filled. In order to store, the bladder is inhibited from contracting as it is filling with urine. Nervous control of the bladder is autonomic and uses spinal nerves and the spinal cord. As the smooth muscle of the bladder slowly stretches, it is inhibited from contracting. Once the bladder is filled, the smooth muscle is maximally stretched. We then fire the Micturition reflex. This reflex synapses in the brainstem and results in the opening of smooth muscle, internal urethral sphincters, as well as the contraction of the bladder smooth muscle, the detrusor muscle. That's when you get the urge to pee. Thankfully, your somatic nervous system controls external urethral sphincters made of skeletal muscle, allowing you to wait until it's convenient. This is one of the few reflexes that can be ignored for a few minutes, and we've all had that experience. You need to pee, but you're really busy, and you forget for about 20 or 30 minutes, but the urge comes back. You can usually get about 3 cycles before you can't take it anymore. Oh, by the way, that's the end of lecture.